So in this lecture, we will be talking about mechanisms, the way an enzyme catalyzes a reaction, converting substrate to product. So these are the different types of mechanisms that we will go over during this lecture. A lot of review of organic chemistry, without a doubt. So we want to know the major players of each of these mechanisms. The enzymes that we will be talking about, whose mechanisms we will elucidate, are RNAase A, serine proteases, which is a broad class of different proteases that use serine. Carbonic anhydrase, which we've learned in the chapter involving hemoglobin, is important in blood buffering. There's about 12 different forms of carbonic anhydrase. And then lysozyme. Lysozyme is an enzyme that breaks apart the cell wall component known as peptidoglycan. If you remember, we studied peptidoglycan in the module concerning lipids and carbohydrates. Before we go into these enzymatic mechanisms, just want to give you some main points here with regards to pH and temperature. Enzymes will operate at an optimal pH. They will also operate at an optimal temperature. So here's an enzyme fumarase that is important in the Krebs cycle. Its optimal pH is around physiologic pH. Well, some other enzymes have different optimal pHs. Trypsin, which is a protease, can cleave peptide bonds, has an optimal pH below physiologic pH. The point here is that, remember, enzymes are proteins. They'll fold into a three-dimensional structure-function paradigm. So there's an optimal pH where it could function, hopefully, and mostly it's physiologic pH. There's also an optimal temperature. So that fold, that three-dimensional fold, that tertiary structure, works optimally at the right pH and temperature. You can imagine if you lower the pH, make it more acidic, you proteinate the enzyme, help it denature. If you make the pH very high, very basic, you'll deproteinate certain amino acids in the enzyme. And also, if you raise the temperature of an enzyme, it's going to denature as well. So main point here, optimal pH, optimal temperature for an enzyme. So our first enzyme that we will study the mechanism of is RNAase A. RNAase A or RNAase S, it's just a different version of the enzyme. Whatever it is, RNAase A or RNAase S, what it does is it cleaves RNA. So enzymes usually end in ASE, and this one has the function of breaking apart an RNA molecule. It's literally scissors, and it's going to cut RNA. So here is an RNA strand, and it's going to cut at this phosphodiester bond right here. It's called a phosphodiester bond. So the active site that's going to participate in this are two histidine residues. So RNAase A is going to function through the chemistry of two histidine residues. More specifically, it's going to do acid-base catalysis. So here's the mechanism. Now, when I lecture on this, we're really talking about main points here. We're talking about major players, major amino acid residues, and main points. We're not interested in memorizing the whole scheme, or we're not interested in memorizing the electron flow. So with that in mind, let's go over the mechanism of RNAase A. So we have two conserved histidines, histidine 12 and histidine 119. It does not matter the number of the primary sequence, but it's just two histidines. Here's our substrate, the RNA molecule. All of this here's the other amino acids in the protein. Remember, it's got to fold into its correct three-dimensional shape. Also, it's got to fold correctly so that the substrate can fit into the active site and the two histidines can do their job in acid-base catalysis. So the first step here is, as we follow the mechanism, this nitrogen is going to abstract a proton on the number 2 carbon of the RNA molecule. So when it does that, this second molecule will serve as a leaving group. So remember, leaving groups from organic chemistry. So as a result of this nitrogen this imidazole side chain picking up that proton from the number 2 hydrogen, you actually have a cyclic nucleotide. This is a 2,3 cyclic nucleotide. Remember, the strand here has formed a leaving group. So now this cyclic phosphorylated nucleotide is going to undergo a change through the help of water. So a common theme here as we go over the mechanisms of these enzymes is that water is going to come in and play a role. So water is going to enter into the active site. And now here, this second histidine is going to serve as a base. So remember, bases pick up protons. So it's going to pick up a proton from this water. That OH group is going to do nucleophilic attack on this phosphate. This oxygen picks up that hydrogen, and now we have generated the 2 histidines in their original configuration. Okay, this one is protonated, that one is deprotonated, they're side chains. And then the second half of the strand is cleaved, it leaves. So in this mechanism, 2 important things I'd like to tell the class is that, you start off this histidine deprotonated and that histidine protonated, and then as the substrate cycles into its product, you need to regenerate back the original active site so that another RNA molecule can come in. So that's the idea behind enzymes. The mechanism has to reproduce the free enzymatic form ready for a second cycle. Also note here that histidine in this version is picking up a proton. So those that pick up a proton are called bases. And then here in this mechanism, in the 2,3 cyclic phosphorylated nucleotide form, this histidine is actually serving as an acid, and this histidine here is serving as a base, as opposed to here it was serving as an acid. So literally the 2 histidines switch roles as an acid base. So this is acid-base catalysis. The histidines are important, the 2 histidines, and we would expect them to be conserved in all RNAases from all different forms of life. So the overview of the RNase A mechanism is histidines, the 2 histidines, acid-base catalysis, and then the importance of water. So the mechanism of RNase A is an example of an acid-base catalysis. So if you remember, if I go back to the previous slide, the 2,3 cyclic nucleotide intermediate is shown here. Only when water comes into the active site does this intermediate break apart. Our second enzyme whose mechanism we'd like to study is carbonic anhydrase. So carbonic anhydrase is going to it's in equilibrium, so it can really go back and forth. It's going to take CO2 and water and give you carbonic acid. Or vice versa, the carbonic acid can dissociate into CO2 and water. Carbonic anhydrase can go in both directions. So this version of carbonic anhydrase that we're going to be talking about is a version that requires zinc. There's 12 different isoforms of carbonic anhydrase. The one that's found in the eye is actually implicated in glaucoma if it's mutated. But we're going to talk about a generic form of carbonic anhydrase, not the specialized form that's in the eye. But the generic form of carbonic anhydrase has zinc as part of its active site. So remember, metals can play an important role as cofactors in the enzyme catalyzed reaction. So here is the zinc shown here as a sphere. A couple of important amino acids that sort of help coordinate that zinc in the proper place in the active site, and that is 3 histidines as well. So 3 histidines, a threonine here, and glutamic acid actually bring that zinc, sort of anchor it in place. Mechanism of carbonic anhydrase. So that's shown here in this diagram. So what the zinc plus 2 does, and here are the 3 imidazole groups. That's the side chain of histidine, the imidazole group. What the zinc does here is that it makes the water molecule actually more OH-like. It actually tugs and pulls on the water and it tugs and pulls so much, we're talking about the zinc here, it tugs and pulls on this water molecule so much that it liberates an H+. When it liberates an H+, what you have left is an OH-. That's sort of coordinated with one of the zincs. Okay, it's coordinated on 3 other ligands with the histidine side chain, the imidazole group. Water comes into the play one more time. Water cleaves this, and now you have carbonic acid. So this version of carbonic anhydrase is going here from right to left where we're making carbonic acid. If you want to go from left to right, all you do is just reverse the mechanism. So now we will talk about our third mechanism here, and this is lysozyme. So lysozyme as an enzyme actually cuts at N-acetylmuramic acid and N-acetylglucosamine. So if you remember, that's the main polymer of peptidoglycan. So reviewing peptidoglycan, it's the main component of the cell wall of bacteria. Gram-positive have more peptidoglycan layers than gram-negative bacteria. So lysozyme is an enzyme shown here that actually cuts right at this beta-1,4 glycosidic bond. Lysozyme, in doing so, actually requires 2 conserved active sites that do the actual chemistry, aspartic acid and glutamic acid. If you actually line up lysozyme, if you do the pi mole assignment, you'll actually see that if you line up lysozyme from human and chicken lysozyme, the aspartic acids and glutamic acids actually line up perfectly. You can line them up one by one, from hen lysozyme, chicken lysozyme, human lysozyme, mice lysozyme. So the tremendous conservation not only in the structure but also in these 2 amino acids in performing this cleavage of the beta-1,4 glycosidic bond. So one of the things that we want to recognize here is when the substrate goes in, here is our peptidoglycan shown here. The model has it such that at the cut site, so if it's going to cut between site E sugar and site D sugar, remember, N-acetylglucosamine, N-acetylmuramic acid, lysozyme cuts at that beta-1,4 bond. So if it's going to cut at this beta-1,4 bond, the cut site, one of the sugars in the cut site undergoes what's called a half-chair conformation. So a half-chair conformation seems to be very important. Remember, these are sugars here, so they're going to have a cyclohexane ring. So here you see a chair conformation of these sugars, but to get it to actually cut at the cut site, it's going to need a half chair conformation. So here is our chair form of one of the sugars, and what we do in the half chair conformation is we take the oxygen and we push it down. When you push the when you push the oxygen down, automatically 4 atoms become coplanar. C1, O5, C5, and C2. They actually form a plane. Okay, they actually are in a plane. And that accommodation seems to help in the nucleophilic attack by glutamic acid and aspartic acid. So let's go over the mechanism now and see how these 2 amino acids in the active site, very conserved from mice to man in lysozyme, how they cut. And important to this cutting is right at the cut site you have the sugars undergo a half-chair conformation where 4 of the 6 atoms become coplanar. So a very important thing here that I want to mention here with regards to lysozyme, the 2 important amino acids are aspartic acid and glutamic acid. And in hen egg white lysozyme, where the mechanism was first elucidated, that's aspartic acid 52 in the primary amino acid sequence, and glutamic acid 35 in the primary amino acid sequence. But I want to bring your attention right here to the glutamic acid, particularly the side chain. Now, the side chain of glutamic acid has a pKa of roughly around 4. So does the side chain of aspartic acid. Okay, if you go back to your flashcards now, the side chain of aspartic acid also has a pKa of about 4. But let's look very closely at this mechanism here. This side chain of glutamic acid, which has a pKa of about 4, is protonated. However, this side chain of aspartic acid, which also has a pKa of roughly 4, that side chain is deprotonated. So how is it within the confines of this enzyme microenvironment? One side chain is deprotonated, the other side chain is protonated, yet they still have the same pKa's. All right? So again, it's very, very important that an enzyme folds and folds properly. So the folded form of this enzyme sort of stabilizes the aspartic acid pKa so that it's deprotonated. Likewise and similarly, the folded form of the enzyme, again, the environment of the microenvironment of the enzyme close to the active site, stabilizes the glutamic acid side chain so that it's protonated. So you can actually see that the enzyme within the confines of the active site really changes the pKa's, okay, causes pKa shifts. So here we go. The substrate's going to bind. Our substrate here is peptidoglycan, and we're going to focus on the sugar, N-acetylglucosamine and acetylmuramic acid. Okay, we're not going to focus on anything else of the cell wall polymer. So bringing your attention now to the two very critical active site and amino acids that will do the chemistry. Here, glutamic acid, remember it's protonated. Here is aspartic acid or aspartate, remember it's deprotonated. Our substrate's going to bind. And the first thing we actually see here is this oxygen abstracts from the glycosidic bond. It abstracts that proton. Okay, and when it abstracts that proton, this sugar labeled E in your textbook, okay, actually leaves. It serves as a leaving group. All right, so there's that H in green. It was picked up from that side chain of glutamic acid. So really what has happened here is that glutamic acid delivers that acidic proton. Okay, it releases that proton, and by releasing that proton to this oxygen here, it disrupts the glycosidic bond and actually serves this sugar to actually leave. So this sugar leaves along with the rest of the peptidoglycan molecule. Remember, this is a polymer. All right, this is where the half-chair conformation becomes important, because this form here, okay, that's still remaining, that's still bound to the active site, the enzyme substrate complex here, actually undergoes the half-chair. Okay, and you have a transition state that's resonance stabilized. So here you have our carbocation. The carbocation is actually stabilized by resonance. Okay, so anytime you have a resonance stabilization, that's pretty good. Okay, though carbocations, you may remember from organic chemistry, are very reactive. So this sort of stabilizes this transition state here. Well, carbocations, this transition state here, also known as the oxonium ion, is resonance stabilized, but carbocations are so electrophilic. And this aspartate, well, that oxygen deprotonated is so nucleophilic. And so it's just a match made in heaven here, where this oxygen does nucleophilic attack on this carbocation. It's resonance stabilized, but it still will do nucleophilic attack. All right, so literally an enzyme substrate complex. Here's our substrate attached to one of the active site residues of aspartate in lysozyme. Well, a common theme is water coming in. So water, very important for the mechanism of a lot of these enzymes. And here's water coming in. Water is going to do acid-base catalysis. And so in this case, this glutamate, okay, which delivered the proton in the first part of the mechanism, takes that proton back from the water. That OH group attacks this anomeric carbon here, and then this pair of electrons goes back to the oxygen, and then we regenerate the familiar configuration of the active sites glutamic acid and aspartate, and also this part of the sugar leaves. All right, our next mechanism here is serine proteases, and this actually is a combination of all the other mechanisms that we've talked about before. There's a separate video on here, and so I do invite you to look at the video that's exclusively dedicated to this. However, I want to hit through some main points here. One of the main points of serine proteases is the universality. So serine proteases from plants, serine proteases that digest and cleave peptide bonds in our gut, serine proteases in our cell, serine proteases in HIV, serine proteases everywhere are going to utilize this mechanism. And they're going to utilize what is called the catalytic triad, serine, histidine, and aspartic acid. The catalytic triad is key. It's a key feature, a key point in all serine proteases in all domains of life. As we go through the mechanism, and once again, please watch the other video that's more dedicated and focused to this, we're going to undergo two tetrahedral intermediates. It's the serine that's actually going to do the nucleophilic attack, but that serine side chain is going to be made very nucleophilic by the pulling power of histidine and aspartic acid. So a lot of the mechanism that we learned from serine proteases, this is classic biochemistry, classic studies, kind of like the Anfinsen experiment, that's very classical. Mechanisms of serine proteases are also classical stuff. How do we know a lot about serine proteases? We learn a lot about the mechanism of serine proteases through the action of poison, specifically this poison called diisopropyl fluoridate. So this DIPF is something that's very, I think it's used in insecticides, insect killers, and roach sprays. Very nasty chemical. And the mechanism of DIPF, this toxic chemical, I also believe it's a nerve agent. But be that as that may, the DIPF is a suicide inhibitor of serine proteases. And basically what happens is that here's your DIPF. Fluorine is a very good leaving group, and that part of the molecule shown in red locks up that serine, okay, the serine here. A covalent attachment, and then the enzyme is practically dead. The cut site is really determined by the specificity pocket. The nucleophilic attack and the tetrahedral intermediates, okay, that's all in the domain of the three important catalytic triad residues, serine, histidine, and aspartic acid. So a couple of the specificity pockets here. Elastase, trypsin. You see here trypsin likes to cut at lysine, or basic amino acids. Chymotrypsin likes to cut next to a phenylalanine. So they're specificity sites. This slide is very important for two main reasons. Number one, the overall conservation. Okay, so if you do a primary amino acid alignment of 100 serine proteases, the serine, histidine, and aspartic acid will all line up, give or take, but they're all going to line up very, very well. Again, the universality of this mechanism of all serine proteases from plant, bacteria, to man. The second important point here is this catalytic triad, serine, histidine, and aspartic acid. The serine is going to do nucleophilic attack. The histidines and the aspartic acid pull. Okay, so this is a hydrogen bonding pull. And when you pull and tug, it makes that oxygen more nucleophilic so that it can cut the peptide bond. Okay? So this mechanism it's very complicated, and so I'm gonna invite you to look at the separate video for this. But I want to focus your attention here on the serine, histidine, and aspartic acid. So here's the side chain of serine. That's what I'd like for you to focus on. And normally the side chain of serine has that OH group, the hydroxyl group. Again, look here at the histidine. Now we call this the imidazole side chain. Look at nitrogen number 3 and look at that hydrogen bond. So that hydrogen bond is really tugging. Okay, it's not an outright pulling, or it's not an outright extraction of that hydrogen, but it's a really powerful pull. And then I want to focus your attention on the N1 of the imidazole side chain of histidine. That N1 is protonated with a hydrogen, but what is tugging at that hydrogen? Okay, what's tugging at that hydrogen is the aspartate or aspartic acid. So you see that hydrogen bond with the carbonyl C double bond O group. So you have a hydrogen bond pulling this way from aspartate. You have a hydrogen bond pulling this way from the N3 of the side chain of histidine. The net effect of all of this is making that oxygen more and more nucleophilic because you have a tug, you have a pull. What's causing that pull? It's this hydrogen bonding pull from two important amino acid, histidine and aspartate. Histidine and aspartic acid, or histidine and aspartate. To give you an idea of how important and critical the catalytic triad is on the mechanism of serine proteases, if you mutate a serine, well, you kill the enzyme because there is no nucleophilic attack. However, if you kill these ancillary parts of the triad, if you mutate the histidine, you will realize that the serine proteases has a very low Vmax, and the Km is practically obliterated. All right. Even though the histidine is not a major player, the hydrogen bond pull is of major importance for this mechanism. Also of importance, if you mutate the histidine, the serine protease activity goes down. If you mutate the aspartate here, also, along the same vein, the serine protease activity goes down. The Vmax plummets, the K of M goes up. Basically, you have an ill-functioning serine protease. So it's not just the serine that's important, it's the histidine and aspartate, such that if you mutate any one of the 3 amino acids of the triad, the serine protease functionality is essentially destroyed. Key point of this slide is that hydrogen bonding pull. Okay, so the arrow's really pulling this way, making that oxygen more nucleophilic. I want to bring your attention here also to the 2 tetrahedral intermediates, one here and one here. Those tetrahedral intermediates are very unstable, and they tend to leave. So you can see here leaving group forms after the tetrahedral intermediate collapses. And then water comes in again. Our old friend water will come into the active site and sort of regenerate and finish out the mechanism.